It's a pleasure to be here. I'd like to thank the organizers for their invitation. For the interest of the time today, I will go over just label-free lab-on-a-chip devices for EV detection and characterization. And I will cover some of the important technologies that are already out there. However, I won't be able to cover all the notable contribution in this field. So bear with me. I'd like to give some examples from recent literature. And my goal here is to give you a flavor of some of these technologies and their future prospect. So to give a little bit of a background on this field, we want to be able to categorize EVs function in terms of therapeutics or diagnostics. In terms of therapeutics, we usually require to extract, isolate, and detect EVs from large sample volume. But in terms of diagnostics, we are usually interested in developing devices that can extract, isolate, detect, and characterize EVs from a small sample volume. So therefore, there is a need for technologies to detect from different sample types and volume. We are interested to minimize complex sample treatment. So we want to minimize some of the tedious work that we have to do to have more hands-off technologies that can be semi-automated. Also, we like to have fast and robust detection in low-cost manner, and eventually we want to get into the point-of-care type of diagnostics. Another feature that we like to pursue is to have non-invasive EV characterizations for being able to utilize these EVs later on as a therapeutics in the field for translational research. So lab-on-a-chip devices, they have these microfluidic platforms that can actually help with some of these criteria. For example, you can have these microfluidic technologies in order to multiplex and having EVs separated or characterized from a large sample volume. Also, since these devices have very small features close to the physical feature of the EVs, you can actually detect EVs from a small sample volume, so it has a pretty good sensitivity. Some of these label-free detection devices can be categorized based on their signal transduction, based on the optical readout or electrical readout, and I will go over some of the examples in the field. However, before I go into showing some of these examples, I'd like to give you a flavor of why do we care so much about label-free sensors compared to all the other technologies that we heard today about labeling EVs and detecting them with different, very highly sensitive detection methods. Some of the features are EV sensing can be based on their unique biophysical properties compared to the chemical properties, and it can actually have some sort of complementary characteristics of some complementary characteristics of the EVs. Also, I'd like to have the detection of the EVs relying on EVs' native and unperturbed biological conditions. Therefore, we can utilize them later on for therapeutic application. Some of these devices have the capability of real-time quantification of the EVs and reduction of analysis complexity and time, and minimizing some of the background signal. As you heard, when we are having multiple antibodies or aptamers, then we are introducing some sort of complexity and background noise into the system. Therefore, we like to minimize all of those. So one of the optical readouts and one of the sensors that has a very powerful feature is surface-enhanced Raman spectroscopy. As you can see, detection of the EVs can happen in both dry state and liquid state when we are utilizing this technology, and it can go all the way down to single EV detection. So the way it works when we are having when light is interacting with the molecule on the surface of these plasmonic surfaces, it will gain or lose some energy. And the changes of this energy depends on the vibrational rotation mode of these particles or molecules. Therefore, it can give you some very specific information about molecular cargoes of the EVs or surface protein of the EVs. I'd like to highlight some of these new technologies that actually utilize machine learning and utilizing this on-chip SERS technology for detection of the EVs as a biomarkers for early cancer diagnostics from 6 different cancer types simultaneously. So this machine learning not only help with detection, classification and detection of these pathogenic EVs, but also could map these EVs back to their cellular origins or tissue of the origin. Other contribution in this field is coming from Randy Carney group, for example, that they can detect ovarian cancer cell-derived EVs with limit of detection of 600 EVs per milliliter. Others show for 97.4% accuracy of EVs coming secreted from prostate or colorectal cancer cell line, and some other technology, some other groups show that EVs can be distinguished from 2 glioblastoma cell lines. So it's a very powerful method for detection of the EVs in a label-free manner. Another optical readout is surface plasma resonance, which depends on the plasmonic method. And basically, it depends on the cumulative oscillation of the electrons between, actually, can I? Yeah. Between the gold surface or plasmonic surface and the dielectric media. When we are shining light into the prism onto this plasmonic surface, we can actually get a detection as the reflective index of these light is changing. So the angle is changing as the particles are binding to the antibodies that are functionalized on the surface. As the refractive index actually changing, we get a change in the angle of reflective light. One of the early work that has been utilizing this technology for detection of the EV is from Im et al. That they showed that they could utilize these nanoplasmonic nanoholes for detection of the EVs from the biofluid. Another work from the Matt Rao group demonstrated that they could detect HER2 positive EVs compared to from the solution of other EVs and microvesicles utilizing device. With a detection limit of around 8,000 EVs per microliter. Another feature of this technology is that it can actually report the dynamic interaction of EVs and antibodies at a single EV resolution. Therefore, it can give some very important information about EV antibody binding kinetics. Others also demonstrated that they showed that they could actually detect 2 specific biomarkers on the exosomes that related to the progression of the glioma. So it's a very powerful tool with a very good limit of detection. Besides optical readout, we are interested in electrical readout as a powerful label-free tool for EV detection. For example, one example is here is a field effect transistor. And the way this device works is basically we have a silicon nanowire or graphene substrate in between the source and drain of this transistor. And as we are functionalizing these surfaces with antibodies and particles or EVs, are binding to the surface, we will get an increasing current or decrease in the current. Depends on the charge of the particle. So when we are having an N-type transistor and we have a positively charged molecules, we are accumulating some holes in the nanowire, and then also we are increasing the current that is passing through between the source and drain, and vice versa. So this is a very interesting and powerful tool, and Klein group at Imperial College London demonstrated that they could actually modify the surface of these field effect transistor by adding some carbon nanodots on the surface in order to increase the electrical double layer of these devices in order to have a better sensitivity and capturing these EVs and being able to show the flow of the current into the transistor. So some of the limits of detection that has been reported is 100 particles per microliter for EVs. Some other groups demonstrated 33 particles per microliter, and also it can be integrated with microfluidic systems for point-of-care type of diagnostics. Another electrical readout is electrochemical impedance spectroscopy. This work has been done by Dr. Lee at MGH Mass General Hospital at Boston. So what they have done is they're having these gold electrodes and they're functionalizing them with antibodies. And as the EVs are binding on the surface of the electrodes, they will be able to measure the change in the impedance of the system. So they have demonstrated that at 10 hertz, they get the best resolution for detection of the EVs, and they were going down to 500 EVs per ml. My group at University of Cincinnati also contributed into this field by developing a new electrical impedance spectroscopy. The way this system works, basically we are measuring or characterizing the unique dielectric properties of the EVs utilizing the electrical impedance spectroscopy. So the electrical impedance spectroscopy is based on the Maxwell mixer theory for analysis of the dielectric properties of the EVs. The way it works, when we are applying a low AC field at wide range of frequency, we can characterize the particles based on their size. At lower range of frequency, at intermediate range of frequency, we can characterize them based on their membrane capacitance, which basically for our EVs is their membrane, lipid membrane composition and the proteins that are embedded on the membrane. And at higher range of frequency, we can actually tap into their cytosol and we can characterize them based on the charge in their lumen. So this is a lab-on-a-chip device, the last revision of this device, where we have a series of glass nanopipettes and 2 sets of microelectrodes for first trapping the vesicles utilizing a DEP force, and then after that we are measuring the impedance of the trapped nanovesicles utilizing these microelectrodes that were fabricated across these nanopipettes. So the video here shows that, is it playing? Oh yeah. So the video here is the microscopic images of these nanopipettes showing that with correct polarity of the voltage, we are inducing 3 different electrokinetic forces that are actually balancing at the tip of the pore and creating this trapping zone in order to immobilize the EVs at the tip of the pore. We have a few different publications to show that actually the yield of our DEP device is comparable to the differential ultracentrifugation. Actually, we have a higher yield and the integrity of the EVs are intact. Later on, we utilize this measurement for impedance spectroscopy, and then we are applying AC field at a wide range of frequency in order to characterize EVs that are coming from different cancer cell types and stem cell, and we demonstrated that at intermediate to higher range of frequency, we could characterize and distinguish between these EVs. Basically, this provide information about their membrane composition and inner lumen charge distribution. And the detection limit of our system is around 1,000 EVs per ml. Another notable data that I like to share here is that, as you know, LDL, lipoproteins, and small EVs having a similar size and density, so it's very difficult to distinguish between them. But they have a different composition in terms of the membrane and in terms of the inner charge molecules that are carrying inside. So we wanted to show that our impedance spectroscopy actually can distinguish between them, and we demonstrated that intermediate to higher range of frequency, we could actually distinguish between these 2 nanoparticles. So to give you some pros and cons of these label-free lab-on-a-chip devices, this review paper very nicely summarize all the pros and cons of these technologies. For example, with CERS and EIS technology, we can actually discover EVs biomarkers based on their lumen biomolecules and surface protein. It gives a fingerprinting response and signal, and we can have machine learning and AI in order to analyze the data. But in terms of utilizing CERS, SPR, and FET, basically surface plasma resonance, and field effect transistor, we need to immobilize these EVs on the surface, and only they provide information about the surface protein of the EVs. However, they have a very good limit of detection, and we can do multiplexing for EVs profiling. Some rigor and reproducibility. We're following the MICEF guideline that was published in 2023. So basically, we're required to report all the instrumentation, any software, any code that we are developing for these devices. And it's very important to actually report the limit of detection of these technologies for the future references. On the horizon, I think we're going to have a multidisciplinary perspective. We will have label-free detection with multiplexed signal analysis utilizing AI or machine learning. Also, we are going to have, I think, real-time longitudinal detection of the EVs. And hopefully, at some point, we can characterize EVs based on their unique biogenesis. And another thing in the future, I believe that we are going to be able to monitor phenotypic changes of EVs over time, similar to the new published work by Matra group that shows that they could actually track extracellular vesicle phenotypes after the treatment of the cancer. And he's going to be one of our plenary speakers here this week, so please don't miss his talk. With that, I'd like to thank you for your attention, and we're happy to answer any question later on in the panel. Thank you.